if you open up a book on evolution and cooperation, you will almost always see an example that's given that's along these lines. And I don't think you're going to be able to hear this guy. Let me see here. Okay. Right, so that is a Belding's ground squirrel, and it's giving an alarm call. And the whole idea is that there's all kinds of nice empirical evidence that giving that alarm call is really dangerous. And it benefits everybody else because all the other squirrels around there are running for safety. And so it's a great example of altruism and cooperation. The individual who does the altruistic act pays some cost. It's really costly to stand up in the top on your hind legs in a field and scream as loud as you can when there's a predator around, but it benefits everybody else. Right? And so from an evolutionary perspective, that's where the riddle is. The riddle is, how can you ever get the evolution of a behavior that benefits others, but it's costly to the individual who takes it? I'm going to talk a lot more about that specific general issue tonight when I talk at the Pilar Institute. But today what I want to do is describe to you some of the experimental work we've been doing on this. But instead of using ground squirrels or something large and furry and warm like that, over the last 10 to 15 years, there have been a number of really beautiful microbial systems that people have looked and used the evolution of, and studied the evolution of cooperation in. So I'm going to tell you about some experimental work that we've been doing on cooperation and altruism in E. coli. Okay? And as I go through this, keep in mind again that when evolutionary biologists talk about altruism, what we mean is that an individual pays a cost for doing the altruistic act, but it benefits others. Okay? So just like the ground squirrel case, that same thing is going to apply in the E. coli system. And then we're going, and I'll explain to you how, and then we'll walk through a whole bunch of different experiments to allow us to sort of tinker experimentally with the evolution of cooperation. So what we've got are E. coli cells that a colleague of ours by the name of Richard Lenski, who has been doing a 60,000 generation experiment in E. coli, he sent us some strains of E. coli. And typically, E. coli, when they're resistant to some antibiotic that we use, right, the reason that they are resistant is that they have a gene or a set of genes on a plasmid that protects them against certain antibiotics like ampicillin. Okay, so they break down the, okay, right. So they're resistant to antibiotics because they produce this substance called beta-lactamase. And beta-lactamase breaks down antibiotics. And that's typically, if a cell has it, they're safe. If they don't have it and they can't produce this beta-lactamase and you put antibiotics around, then they're dead. Okay. And like I said, these are typically associated with, the genes are typically on plasmids. Now, the way it usually works is if you produce beta-lactamase, you keep all of the beta-lactamase that you produce. And so if there's something dangerous around, you're safe. If there's antibiotics around, you're safe. Now, with our E. coli, what we did was we experimentally created strains of E. coli that share some of the beta-lactamase that they produce. Okay. So we engineered, in our case, E. coli cells that were identical to the typical E. coli cell, except what they did was they kept some of the beta-lactamase that protects them against antibiotics, but they secreted out into their environment some of the beta-lactamase that protects everybody against antibiotics. E. coli doesn't usually do this, but lots of microbes do, in fact, take some of the beta-lactamase they produce and secrete it out into the environment. There is a sense, then, in which these cells that are secreting beta-lactamase are like altruists. Okay? And the reason is that I'll show you in a minute, experimentally, that it's very costly to produce beta-lactamase. It costs the cell something to do that. Some of these cells, the ones that we've experimentally engineered, they take the beta-lactamase and they pump it out into the environment that they're in. What that means is, if there are other cells that are out there that aren't protected by themselves, let's say they don't have the genes associated with producing beta-lactamase, all of a sudden, if they happen to be around some of these cells that are secreting some of the altruists, they're safe in the presence of antibiotics. Antibiotics are like the predator here, right? So if you've got some of these altruists around, it's costly to be an altruist. It protects others in your population. And so it's just like the ground squirrel case, except now you can do all sorts of detailed experimental experiments where you can run multi-generation experiments on the span of weeks rather than years. And you can look at the evolution in real time of altruism in this system. So what we've got are one group of cells that secrete out their beta-lactamase, like I've been mentioning. So they keep some of it and they ship some of it out. We, in fact, have experimentally 4 different groups that I'm going to show you competitions between. So I just want to walk you through them so that when we do the experiments, you can follow what I'm saying. We have one group of E. coli cells, which are just the classic E. coli cells, and those individuals produce beta-lactamase, and they keep it. Okay? So they don't protect anybody else, but they do protect themselves. Then we have the strain we've just been talking about. They secrete some of the beta-lactamase out, but they keep some of it. Okay? Those are our altruists. Then we have some cells that we've experimentally removed the plasmid that houses the genes that produce beta-lactamase. So now they are totally unprotected. Okay? They don't have the plasmid, they don't have the right genes for this, and if antibiotics are around, if the predator is there, then they're in big trouble because they have no protection whatsoever. I refer to those individuals as cheaters because they potentially can be protected from danger without paying any costs if there are altruists around. If there's no altruists around, they're all dead. But if there are these altruists around, then they can be protected without paying any costs for protection. And then we have one final strain that I'm referring to here as the dummy plasmid strain. Okay, and I'll tell you what it is, and then when we do the experiments, it'll be much clearer why we would want this kind of group. What we've done here is we've experimentally removed the plasmid that is associated with the protection, the beta-lactamase. So we've experimentally removed it, just like we did in the cheater strain, but now we've put back in another plasmid that is completely functionless, but we're putting it back in to our E. coli. So they have to carry a plasmid around that's the normal size of a plasmid that they would usually have for protecting themselves, except they're going to get no protection. So they have to pay the cost for carrying around the machinery, but they're not getting any protection. Right? And we basically put all of our cells into an experimental setup like this, where all we have here is a system that allows us to keep giving them nutrients and keep our strains alive and allow competition experiments. And we're also removing all the waste and other things that we don't want. So, but the reason I put this up here also is that this is all done in liquid. So we are constantly mixing up our cells together so that we don't have any kind of population structure. Everybody is mixed around, so we basically have individual cells randomly interacting. It's not like putting them on a plate where you have real strong spatial structure. In our case, there's no spatial structure. All right, so now I'm going to start the experiments after this slide. One last time, 4 different strains. The selfish strain, if there's antibiotics around, they're fine. They survive. The altruistic strain, if they're grown by themselves and there are antibiotics around, they're fine. If you grow the cheater strain by itself, it has that plasmid that's been experimentally removed, they die if they're by themselves and you put antibiotics around. They're dead, unless they're altruists, but by themselves, they're dead. Same thing for the dummy strain. If you raise them by themselves, no other kinds of cells around, and you put antibiotics around, they're dead. Okay, so now what can you do with this kind of experimental setup once you have it? All right. So I tell you a set of experiments that we did where we're testing a lot of the assumptions that people make when they study the evolution of altruism, and then we're also going to see whether or not we can actually have altruism emerge and be maintained in our population of E. coli cells for a real period of time. Okay. So the first hypothesis I'm going to test goes like this. All along, I've been saying it's costly to have to produce beta-lactamase to protect yourself or to protect others. There's some cost that you have to pay for having that plasmid and for producing the beta-lactamase, right? Okay. If that's true, right, then in my system, that cost is paid by any of the cells that have either the real plasmid or the dummy plasmid. But that cost, okay, is not paid by the cheater types because we've experimentally pulled that plasmid out and we haven't put anything back in its place. Okay. So if you take the selfish, altruistic, and dummy strains, all of which have to pay this cost, and you compete them in any way against each other, they should all do approximately as good when there's no danger around, when there's no antibiotics present. So the first thing we did was just ask whether or not we were right about that. And what I'm showing you here is a 7-day experiment, which is many, many generations in E. coli. And in this case, what we've done is we started out our experiment with half of the cells being selfish, meaning that they protect themselves and nobody else, and half of them being the dummy strain. Both those strains carry a plasmid. So they should compete with each other equally well when there's no antibiotics around. There's no danger. They both pay the cost. They should do approximately equally well. You start them at 50% of the population each, and what you can see here is as you run that experiment for 7 days, which again, I don't know is on the order of hundreds or dozens to hundreds of E. coli generations, and it stays right around that 50% level, suggesting that they are in fact equal competitors with each other when there's no danger present. And the same thing holds true when you compete the dummy strain, which has this fake plasmid that doesn't do anything, it doesn't provide any protection, but it's there. And you compete them against the altruist that has the plasmid. Again, when there's no danger, when there's no ampicillin around, and you start them at 50-50, they stay there essentially. All right. The cheater strain that we pulled the plasmid out of, it doesn't pay that cost. So it should do better than our other strains when there's no danger. If there's a danger, then all of a sudden not having the plasmid is important. But if there's no danger, then you should be able to do better than everybody else if I'm right that having the plasmid is costly. What we have here is, again, in this case, a 5-day experiment. And what we're competing in the darker line, which is the line on top, is the altruists versus the cheaters. They have no plasmid, the cheaters. And what you're seeing is that the frequency of the altruistic strain is going down. If you compete selfish types, which are the lighter circle, the lighter lined, okay, and you compete them against cheaters, then again, the selfish types have the plasmid. The cheaters don't. Cheaters should do better. If you look at that bottom line, the lighter line, what you're seeing is the frequency of the selfish types going down. So the y-axis is either the frequency of the altruists, which is the top line, or the frequency of the selfish types, which is the bottom line. They're both going down when they're competing against cheaters. Cheaters do better when there's no danger. This is a classic assumption people make when they study the evolution of altruism. It's very hard to test. But in this system, it's easy. So it's costly to do this, to protect yourself, and yet others get the benefit as well. So how does that work? Now that we see experimentally that all of the assumptions that usually one makes when you study this sort of thing seem to be verified in our system, I want to show you an experiment in which we compete altruists and other types of cells with each other when there really is danger. Everything else up to now has been no danger. But what if the danger is there? What if there are antibiotics? Before I show you the experiment, I want to walk you through a very, very basic idea on what it is that we expect to see. Theoretically, what do we expect to see? So I'm going to show you just a very simple model that we devised. And on the x-axis is the frequency of altruists. These are the frequency of the cells that are protecting others. And on the y-axis is fitness. And this is a simple graphical model, okay? Now, we know from our work and from the work of others that generally speaking, you need to have at least a few altruists around before altruism really kicks into effect. So in a system like ours, it's not enough to have one E. coli cell who's an altruist. You need to have some minimal frequency of altruists before they really can protect others in a system like this. So I'm going to call whatever that is, this value P min, which is sort of the minimal frequency of altruists you need so that other individuals are protected. So if that's the case, then this simple line represents the fitness of cheaters, those who don't pay the cost. After P min, their frequency should start increasing linearly like you see here. If it's below P min, there's not enough altruists around to protect anybody. If it's above P min, the more altruists are around, the better it is for you as a cheater, the more altruism that's being tossed out there for you to cheat on. And we can follow that line and see that it tops out at this value B, which stands for benefit. This is the maximum benefit any individual in this system could have. So we basically have the fitness of cheaters represented that way. Now, we can also graph out the fitness of altruists, which I've shown in red here. Now, altruists in our system also protect themselves. So they're protecting others, but they also protect themselves. So even when the frequency of altruists is very low, altruism fitness is fine. Right? Then, the more altruists around, the better. But it is going to cap at a lower value than where the fitness of cheaters capped. Because by definition, the altruists have to pay this cost of being altruistic. So the highest fitness value they could get is the benefit of having altruists around minus the cost of having to be an altruist and pay that cost. So their fitness maxes out at B minus C. If everybody's an altruist, then everybody gets this benefit minus the cost. But if everybody's an altruist and you're a cheater, you get the whole benefit and you don't have to pay the cost. So this is a very, very barbarically simple way to kind of graph out the fitness of cheaters and altruists in a system like ours. if you want to know what to expect when you compete altruists and cheaters, you look for the equilibrium value. And that is just where the lines cross. And I'm representing that by P star. So that's the equilibrium frequency of altruists. Right? So if you tilt the lines differently, you're going to get a different equilibrium frequency. But for these two lines right here, that's the expected value, wherever those lines crossed. Okay? And I'm not going to bore you with the details, but you can algebraically figure out exactly what that value is if you know something about the costs and benefits in your system. So you can come up with a numerical value as well as a graphical solution. But all I want to get across here is that you expect to see some combination of altruists and cheaters. Here, it's a P star is where it is. P star might be over to the left if the lines were tilted slightly differently. It might be over to the right even more if the lines were tilted slightly differently. But for this kind of system, we expect some sort of mixture of altruists and selfish types. Okay, so what happens when you actually experimentally look to see if that's what you get? So I'm going to show you an experiment, and now danger is present. And the danger in our case is the ampicillin. It's the antibiotic that kills these cells. It's what we like to use to get rid of them. What you're seeing here is the results of an 8-day experiment. And at the start, we had a population of E. coli cells. Half of them were the altruists, and half of them were the dummy cells. Now remember, the dummy cells have this plasmid that doesn't do anything. It's the same size as a plasmid that usually provides protection, but it doesn't do anything. Which means that if you take dummy cells and you raise them by themselves in the presence of this danger, this ampicillin, they all die. We did this dozens of times. Every single time, if you run the dummy cells, and only dummy cells, in the presence of ampicillin, they all die. But if you have a population like this one, where half of them start off as dummy cells and half of them are altruists, then you can follow what happens. After a couple of days, the frequency of altruists goes up to about 95% of the cells. And it stays there. About 5% of the cells are dummy cells. Those cells would be dead if there were no altruists around. But you can run this experiment out longer. We've run it for 30 days, and this frequency of 5% stays around there. So you now have a system where you have altruists, even though they're in the vast majority, but you also have these cheaters who are sticking around in this population when normally they would be dead if they had no altruists around them. So you can get the stable existence of altruists and cheaters in a microbial system like this. Now, the nice thing about this kind of system is you can now change the costs and benefits slightly and make a very clear prediction that you can test. And that would be something like this. When we compete altruists and dummy types in this experiment, we get 95% of the cells being altruists and 5% being dummy. Now what happens if I take altruists and I compete them against selfish types? The selfish types, remember, against the cheaters. The cheaters have no protection. Their plasmid has been pulled out of them. They'll die if they're by themselves, just like the dummy cells would die by themselves. But now, if I take these cheaters and I compete them against altruists, they should do better than the dummy types did against the altruists, because the dummy types carry around this plasmid that has no function. It usually provides protection, but we've made it so it's functionless. The cheaters don't have to carry that around. We've experimentally pulled it out of them. So they should do better than the dummy types did against the altruists, because they don't have to pay the cost that the dummy types have to pay. So now we can test that prediction, that if we compete the cheaters against the altruists, they should do better than the dummy types did against the altruists. What you're seeing here is the result of that experiment and also the result of the experiment I just showed you a minute ago. The darker line is the frequency of cheaters when they're with altruists, and the lighter line is the frequency of the dummy types. It's the same line as we just saw a minute ago, about 5% of the cells. You get about 12.5% of the cells being selfish, but you only had 5% of them being altruists when they were competing against, 5% of them being the dummy. You get 5% dummy cells, but you get 12.5% cheaters. 2.5 times as many cheaters. This gives you an estimate of how costly it is to carry around this plasmid that's typically associated with protecting yourself. If you don't have it, if you're a cheater, your frequency is 2.5 times higher, 12.5% here, when you're competing with altruists. So we can manipulate the costs and benefits, make a prediction, and test it. One of the things about this system is, as nice as it is, so far, I've always shown you experiments in which I start off with two types, altruists versus dummies, altruists versus cheaters. And I typically have them at 50% each when I start, and I look at what happens. One reasonable question here would be, would you ever get the evolution of this system if you started off with only one of these types? I know I get some nice mixture of types when I start off with both of them. But would I ever get to that point if I just had a single strain? Would I evolutionarily ever get to the point where I had, say, selfish types and altruistic types and cheaters? I don't know, because in these experiments, I've always started off with both of them. So we ran a follow-up experiment. We ran this 4 times, and here's what we did. We started off with a population that was 100% altruist, these cells that secrete out some of their beta-lactamase. And we wanted to ask, would we naturally, without any manipulation, end up with some cheaters, and would they mix together and equilibrate at the same place as they did in our other experiments where we started off with both types? So basically the question is, if you start off with one, do you evolve into a system with multiple types? It's much easier to do it this way, by starting off with 100% altruists, because typically E. coli cells and most microbes, every once in a while when they divide and reproduce, they lose a plasmid. So it's possible that just naturally, on rare occasions, when they're dividing, they might drop the plasmid that protects them. As soon as they drop that plasmid, they have naturally become the cheater that we experimentally created before. It would be almost impossible to do the experiment the other way. You could start off with 100% of cells that were cheaters and hope that magically you get just the right series of mutations that create an altruist, but the odds of that are trivial in the time frame that we had to work with. So we started with everybody being an altruist, and we want to see if we naturally would get cheaters. So on the y-axis here, you have the frequency of cells that have no plasmid. They're the cheaters. On the x-axis, you have the almost 30 days that we ran this experiment. And here's what you find. If you track the experiment for the first 10 days, you get very discouraged, because every single cell in all 4 of the replicates remains this altruistic secretor type, just like it started out with. Right? But then, in 2 of the strains, one or a very small number of the altruistic cells dropped the plasmid that provides the protection. And as soon as that happened, they naturally became cheaters. That's what cheaters are. They're the cells that no longer have to have that plasmid and the cost associated with it. And then you can watch in those 2 replicates what happens? And what you see is that in both of them, the frequency of the cheaters begins to rise, and in both cases, it seems to stabilize at that value you see on day 27. And if you run this a little bit longer, again, you find some variability, but it tends to stabilize around there. Both of the replicates in which this happened stabilized at the same endpoint. So naturally, you could get this system to evolve because we had 100% altruists. When we're done, we have 2 of our replicates having both cheaters and altruists. From our perspective, what was really interesting was that if you look at the frequency of where the cheaters stabilized, it's at about 10%. About 10% of the cells were cheaters. Right? That's really close to the 12.5% that we saw when we started an experiment where both types were present. What this says is that that's a really robust finding. If you start off the population half and half, you end up with about 12% cheaters and 88% altruists. If you start the population out at 100% altruists, you end up with about 85 to 88 to 90% altruists and around 10% cheaters. So you've got this system now that experimentally you can manipulate variables and test ideas about the evolution of altruism. So let me just ask you guys, I've got about 15 minutes more where I can talk about a second experiment. Should I do that, or should I, okay, would you, keep going, okay. All right, and then we can take a break for a few minutes and we can do the second talk, which is on something completely different, which is how humans and non-humans choose their mates and how genes and culture determine that. So if you're finding this a little dry, I promise you the other one you won't find this dry. But so let's keep going with this. So in our E. coli system, you can get the evolution of altruism, and you can manipulate it and test predictions. Okay, the thing about it is that when you get microbial altruism like this, you can start thinking about the community level effects of altruism, the effects on other species. Because think about it like this, right? A lot of the E. coli, or whatever microbe you're looking at, a lot of them are residing in the guts, in the stomachs and intestines of things like us and other mammals. And when they're in there with tons of other microbial species. Okay? Now, the kind of altruism that our E. coli are doing, where they're secreting out some of this stuff that breaks down antibiotics, that should protect other microbial species just as well as it protects other E. coli. If you're a different microbial species and you happen to be around an E. coli that's an altruist, you're safe because all the E. coli does is just pump this stuff out and it breaks down antibiotics, and that helps everybody, not just other E. coli. So we started to think about an experimental way to look at the community level effects of altruism. And what we did was we constructed the smallest possible community, which is a community made of 2 species, our E. coli cells and a strain of salmonella. Okay. And our simplest question went like this. Given the way that E. coli altruism works, it should be the case that if you're a salmonella cell and you're around one of these E. coli altruists, you should survive perfectly well. Now, the thing about our salmonella strains is they themselves are not protected against the antibiotic we're using. So if you take our salmonella strains and you just raise them up by themselves and you put this danger in, ampicillin, they're all dead. They can't protect themselves in any way. So we thought, okay, now let's take them, let's put them with our E. coli altruists and see what happens. It should be that they survive because all the E. coli does is just produce this public good, this thing in the environment that breaks down the danger. So we ran an experiment, and now at the start of the experiment, you're looking at the percentage of the cells that were salmonella. So 50% of the cells were salmonella, and 50% of the cells were our altruistic E. coli. And what you see is that not only do the salmonella survive, they do really, really well. By the time the 48-hour experiment is over, now 80% of those cells that are present are salmonella. It started at 50%, it went up to 80%, remembering that if there were no E. coli around, there would be 0 salmonella cells. So what this result simply tells us is, it tells us two things. First of all, it tells us that E. coli is protecting salmonella, because they'd be dead otherwise. And it also tells us that salmonella are really good competitors against E. coli. Because not only do salmonella survive, they outcompete E. coli, and they make up at the end about 80% of the population. So there clearly are, in our system, community-level effects of altruism. That was interesting to us. And just for completeness, we decided to sort of run a whole bunch of other competition experiments between our salmonella and our E. coli. And when we did, we found something that was completely unexpected, that in fact, in the end, might be more interesting than anything that you see here. So let me tell you what we ended up doing. Right? We've got all these E. coli strains that I told you about, so we thought, okay, let's just take salmonella and compete them against our selfish E. coli strains. Now remember, selfish E. coli, they produce this beta-lactamase that protects them, but they don't ever secrete it. So all they do is protect themselves, but they don't protect anybody else. So we put them together with salmonella, and our assumption here was that when we do this, salmonella is going to all die, because the E. coli selfish types don't protect anybody. But we thought we better do it for completeness. And we did it, and we got the same basic outcome as we got when we competed salmonella with our altruistic types. We started them off slightly different frequencies, but at the end, about 80% of our cells were salmonella. And this really surprised us, because we did not expect that they would survive in the presence of selfish E. coli. If you take those salmonella strains out, and you just look at the salmonella alone, and you put ampicillin in, the antibiotic, they're all dead. But if you take them and you compete them against selfish E. coli, they seem to do really well. And so we thought, what could possibly be going on here? And we began to run through a number of alternatives. And I want to walk you through those experiments for 5 or 10 minutes, because at the end, we're left with a bit of a riddle, but the possibility of something extremely interesting going on here. So let me walk you through this. Okay? So we thought, how is it possible that selfish E. coli, that are only supposed to protect themselves, seem to be protecting our salmonella? How is that possible? Okay, so we ran through a whole series of hypotheses. Our first hypothesis is that somehow we're wrong, that this experiment was run after the E. coli experiments I talked about, so maybe something strange happened, and those E. coli cells that were selfish strains when we just did the E. coli experiment, since this experiment was months later, maybe they've mutated and they're actually secreting out some of their beta-lactamase. It's possible. So maybe the selfish strain is not selfish anymore. They were in our E. coli experiments, but maybe something's happened and they're not here. Maybe they leak this beta-lactamase out. So there's actually a very, very easy way to test that. What we did was we took these E. coli selfish types and we put them in the presence of E. coli cells that were unprotected. Right? We put them in the presence of E. coli cells that had no plasmid to protect them. When you do that, the E. coli cells that had no plasmid all die. Right? So if our selfish E. coli strain was not selfish, if we were wrong, and it was actually now secreting out something and helping others, then the E. coli cells that were unprotected should have been protected by those cells, but they weren't. They all died. So it's not simply that the E. coli selfish types have become E. coli altruists because of some strange set of circumstances in the months between our experiments. Right? So we ran another possibility, an experiment for another possibility. Salmonella and E. coli are phylogenetically pretty close to each other. They swap genes and plasmids all the time, genes in particular. Okay? So it's possible that Salmonella picked up the genetic machinery from horizontal, what's known as horizontal or lateral gene transfer. They picked up the right machinery from E. coli, and they're simply able to protect themselves. This kind of thing happens all the time in closely related species of microbes. And if that was going on, then it wouldn't be that E. coli were protecting them in any real way. It's just that they picked up the genetic machinery from these E. coli. Right? So there's the first experiment we did, where we just showed, hey, you take these selfish E. coli, you compete them against Salmonella, and the Salmonella will do really well. Right? Then we take out the Salmonella that survived at the end of that experiment, the 80% of the cells that were Salmonella. And we put them in the presence of ampicillin, but no E. coli. And when you do that, they all die. So if they had picked up the genetic machinery, then they don't need the selfish E. coli around anymore. But that's clearly not the case, because if you just raise them in the presence of ampicillin but no selfish E. coli, they all die. Every one of them. So we came up with another explanation. How is it possible that our selfish types are protecting Salmonella? They shouldn't be doing that. You saw that when you put these guys together, Salmonella does really well. They're just really good at outcompeting E. coli for resources. Now, in this system, the protection from beta-lactamase that we've been talking about is when a cell that's living secretes out some of the beta-lactamase and protects others. But beta-lactamase also gets into the environment when cells that have beta-lactamase die, and the cell lyses open, it rips open, and beta-lactamase can get out into the environment. So maybe what's going on here is this. Maybe Salmonella are such good competitors against E. coli that they're killing them all the time, constantly. Those dead E. coli cells are lysing open, and a lot of their beta-lactamase is going into the environment, and that's what's protecting the Salmonella. It turns out there's a very easy way to test that, and that is that when the beta-lactamase would come out of a ripped open dead cell, all the other junk in that dead, ripped open cell would also be in our environment. So we can look to see whether or not there's lots of other stuff that would be in there that comes out of a ripped open cell. And that's not what's going on. The beta-lactamase that's in the environment is not the result of dead cells being ripped open and having the beta-lactamase come out of them. Because if it was, we would see all the other stuff from those ripped open cells that we don't see. Okay. Now, 5 more minutes and we'll be done. Here's where we said, okay, maybe we're thinking about altruism a little bit in much too narrow a way. And we thought that maybe these E. coli selfish cells that we've been talking about, maybe they're not. We know that they haven't just changed evolutionarily into altruists, but it could be that under certain conditions, these selfish E. coli strains begin to act more like altruists. Those conditions were not taking place when we only had E. coli competing against each other, but maybe those conditions that might make a selfish E. coli into a more altruistic E. coli, maybe those conditions are what's going on when we compete them against Salmonella. So let me show you what I mean. Okay. Here's what we know. We know that E. coli cells sometimes produce these things called membrane blebs. All these little projections, my little pointer here is not working, so all those little projections on the outside of the cell, those little bumps, those are what are known as membrane blebs. And E. coli produces those sorts of things under certain conditions. Okay, so they produce these membrane blebs sometimes. Sometimes the membrane blebs have this beta-lactamase that protects you against antibiotics. Sometimes the blebs break off from the E. coli cell into the environment. And we know that one of the things that makes E. coli cells start to produce blebs that sometimes contain beta-lactamase and sometimes break off into the environment, one of the things that makes them do that is when they're in high-stress situations. Okay. For an E. coli cell, high stress means intense competition for resources. And that's what the Salmonella are giving them. The Salmonella are extremely good competitors with E. coli. We saw that from the original results. So it could be that when they're competed against Salmonella, the selfish E. coli strains respond to the stress of competition by producing membrane blebs with beta-lactamase that sometimes break off and go into the environment. The problem that we have is that we do not have the machinery or the money yet to test this hypothesis. So we do not know specifically in our experimental system if our E. coli cells are producing these membrane blebs full of beta-lactamase that break off into the medium. We know that they do all of those things in other experiments, but we don't know specifically if it's going on in our system. But if it was, it would explain what's going on here. The selfish E. coli are selfish, but under certain stressful conditions, they produce these membrane blebs that has the side effect of releasing some beta-lactamase into the environment, and that's how Salmonella are protected. It's possible. But there's one more, and I've told you what we don't know already. There's one more interesting possibility that's related to that. So, if what I just told you is right, it's sort of just an incidental thing that the Salmonella get protected. In other words, they're not doing anything actively to get E. coli to protect them. It's just that they're really good competitors. When E. coli is against really good competitors, they sometimes produce membrane blebs, and that protects the Salmonella. But it's possible that Salmonella is actually actively manipulating E. coli cells that are normally selfish into becoming altruistic cells. So here is a diagram, not by me, but by people who have worked with what are known as type 6 secretion systems. This is a little diagram of this type 6 secretion system that Salmonella is known to have. And essentially what I want you to get out of this is that this is like a tiny little rocket. What happens is, through this type 6 secretion system, Salmonella can actually produce a little piercing rocket-like outcrop that's capable of piercing other cells. If that was happening in our system, then Salmonella might in fact be able to cut a tiny hole in E. coli cells that causes this beta-lactamase to be released into the environment, but not kill the E. coli cell. It's small enough that that's the way it would happen. They could do this without killing the E. coli cell. If they did, they would be actively manipulating a cell that's usually selfish into being altruistic. This is the kind of things we think that maybe primates are capable of, other primates are capable of, not the kind of things that we think of in microbial systems. But we know that Salmonella has these type 6 secretion systems. We know that when these type 6 secretion systems are used, that's what they would do. They would poke a little hole in the E. coli that wouldn't kill it, but might let out some of the beta-lactamase. Like in the last hypothesis I told you about, we simply do not have the resources or the equipment to test this yet. So we're left with two possibilities. Either the membrane bleb hypothesis or this. So, like I said, we don't know. We've already done this, so let me rush to the end point here. We don't know yet, but we're left with two possibilities. Either the shellfish cells respond to the stress of having Salmonella by producing these blebs, which break off into the environment. And that's a kind of passive protection. Salmonella isn't doing anything active to turn E. coli shellfish types into E. coli altruists. The second possibility, the type 6 secretion system, that would turn a shellfish E. coli into an altruistic E. coli by manipulating it, by piercing a hole in it and making it leak out the substance. This raises all sorts of interesting possibilities to people who study the evolution of behavior. So if we could test this, we could do things like this. We could ask something like this. Do the type 6 secretion systems, if that's what's in play here, do they always turn on when Salmonella is in the presence of E. coli? Maybe they do. Or does Salmonella only use the type 6 secretion systems when you have things like shellfish E. coli around? You don't need a type 6 secretion system to protect yourself from danger if you're around altruistic E. coli, because they're naturally doing this. There's some cost to turning on and using the type 6 secretion system. Does Salmonella only do that when they're with types that they have to manipulate, or do they always do it? Again, these are the kinds of questions you tend to think about when you work with big-brained animals. But you have a system here that shows you that kind of manipulation is possible. We don't know if it's happening, but it's possible even in single-celled, simple creatures like this. So our goal in the next few years is to get the money and the machinery to determine whether or not they're producing blebs, whether or not Salmonella in our system is using these type 6 secretions, whether or not Salmonella always uses them or only when it's in their benefit to use them. And we can follow these things up with all sorts of experiments. So I just end by saying there's really a number of different things that this kind of system allows us to do. It allows us to look at the evolutionary implications of altruism in a single species, not only for individuals in that species, but for other species in that community. And we can look at that over ecological, evolutionary time here. This kind of approach also has ecological implications. And the ecological implications are that if altruism in one species protects other species, then all of a sudden altruism has an effect on community level diversity, an ecological question. All of a sudden diversity becomes possible if you have some species that are altruistic where it wasn't possible perhaps without them. Right? If you might have more microbial species present in some community if you have E. coli altruists than if you don't have E. coli altruists, because the E. coli altruists protect everybody. And finally, I would say that there are medical implications here. Now, this is quite far away from my own area of expertise, but I would make the following kind of argument to people in the medical community. There are lots of species that do the kind of altruism that we saw our E. coli cells do. They secrete out something that protects everybody. If you're in the medical community, what you want are antibiotics that your microbes don't get resistant to, right? And you want to get rid of as many possible disease-causing microbes as you can. And sometimes E. coli causes disease. The way we typically think about that is you got to basically get rid of every one of the disease-causing cells in order to get rid of the disease. But if you have a system, a microbial system, where there's some altruism, then we saw that you get both altruistic types and cheater types. And the cheater types have no protection themselves. They only survive because the altruists are around. What that means is, medically, if you targeted the altruistic cells and got rid of them, you would also get rid of the cheater cells, because the cheater cells are dead without the altruists. So if you could target the altruists alone, you could get rid of all of the microbes you're worried about without having to kill specifically on your own every single cell. All you got to do is kill all the altruists. And then as soon as you do that, the unprotected ones, the cheaters, will just die. The cheaters are present because the altruists are present. If you can get rid of the altruists, you get rid of the cheaters without having to target the cheaters. So all in all, I think this is the kind of system that we can really manipulate as an evolutionary biologist, and that has these ecological, evolutionary, and medical implications. I should stop here. Maybe we could take a couple of questions and then take a break and then go back to the next one. Thank you.